Hi, this is Steve Kelly, the CEO of Carisma Therapeutics. Thank you to the organizers for inviting us to participate. I'm going to spend the next few minutes giving you a broad overview of Carisma, what we're working on, and what our development plans are going forward. So Carisma is a 4-year-old company. We were a Penn spinout founded by Mike Klaczynski and Sarah Gill. And the idea behind the company is that we are engineering macrophages for adoptive cell therapy approaches in solid tumors. And the idea came out of Penn. T cells were working in heme malignancy. They had the idea, why don't we try another cell type and see how that works? The challenge, however, was macrophages are incredibly difficult to engineer. And it was only after a year of trying different vectors, AAV and lenti don't work, that they were able to find a vector that could reproducibly transduce macrophages, and that vector is Ad5F35. That was the catalyst to form the company, and over the last 4 years, we've done quite a bit to develop the overall concept. We have a pipeline of emerging agents. CT-0508 is our lead program. It targets HER2. We have just received IND clearance and hope to be in phase one testing very shortly. So maybe actually when this is being viewed, our phase one will be open. In addition to our lead program, we have two other programs, one targeting PSMA, one targeting mesothelin. We'll be nominating development candidates for those, leading to an IND sometime in the second half of next year. In addition to that, we have a very robust discovery engine. We have a number of initiatives looking at enhancing our overall activity, looking at second generation approaches, looking at ways to decrease our overall cost of goods, decrease our vein to vein time, and importantly, look at other applications for engineered macrophages. As we have received an IND, clearly we had to have a GMP supply chain. We've been working on that for the last 2 years. And at this point, we've run over 50 donors through our process. And again, we have good reproducibility, good CAR expression, good cell viability. We partnered with CDMOs, Wuji for our viral vector, and Miltenyi for our cell product. We're currently about 35 employees. We have all functional areas represented. We have discovery, translation, clinical development, manufacturing, process development, quality regulatory, et cetera. And so we have all the functions necessary to drive this program forward. In addition to that, we have great advisors around the table, including Carl June and Sarah Gill, co-inventors of the technology, and Hai Levitsky, former CSO of Juno, is on our SAB as well. There are several other experts in cell therapy and solid tumors that are helping advise the company as we move forward. We have completed a fundraising. As of now, we have one Series A fundraising event, $59 million in 2018, led by AbbVie and HealthCap and joined by a number of other significant investors. And it gave us cash runway into 2021. Now, stepping back a little bit, clearly macrophages are a different cell type from what has been probably the most attention, which are T cells and NK cells, but macrophages themselves are a very powerful cell. They're a member of the innate immune system. They're a first responder. They address any infections. They remove old cells, old protein aggregates through phagocytosis. They're also instrumental in changing the environment around them. And they can be a pro-inflammatory environment producer to an immunosuppressive environmental producer, but based on various cytokine profiles of the phenotype of the macrophage. In addition to that, they can drive the adaptive immune system through antigen presentation, so recruitment and presentation of antigens. They're a tissue resident cell. They're found in, for example, in the liver as Kupffer cells, or the CNS as microglia. They're also found in tumors as tumor-associated macrophages. Those macrophages are an M2 phenotype and dramatically contribute to the immunosuppressive tumor microenvironment and exclusion of other immune effector cells. They're really conducive for the growth of the tumor. Because of that, there's been a lot of attention directed at approaches to look at macrophages and how can we use macrophage biology in cancer. Initial areas of investigation included TAM depletion, to remove that immunosuppressive signal from the tumor microenvironment using CSF1, for example. There's also been a TAM repolarization strategy where you take a TAM and convert it into an M1 macrophage, hoping that it would trigger phagocytosis of the cancer cells nearby in the tumor. There have been blocking of the CD47 SIRP alpha pathway, so remove that don't eat me signal from cancer cells, hoping to engage macrophages. All good and appropriate approaches. However, they lack one thing that we provide, and that is the ability to recognize cancer cells as foreign and need to be eliminated, and then triggering the phagocytosis exercise. And we do that through our CAR. In the paper in the middle there is from August, Nature Biotechnology, talking about our approach. Now, going a little bit more detail on how this works and why we're differentiated versus other approaches, there are three basic issues that other adoptive cell therapy approaches encounter in solid tumors. Number one is tumor access. T cells and other immune effector cells are blocked from entering the tumor microenvironment. So just the access is problematic. Also, if they do get in, there's an immunosuppressive environment, often caused by TAMs or generated by TAMs, that prevent them from having any cell-killing activity. Lastly, there's an issue of antigen presentation. Unlike with heme malignancies, where CD19 is ubiquitous on B cells, there is no similar antigen on solid tumors. And so while you could, if you get T cells to engage, they could kill all of the antigen-bearing cells, but there would be escape for non-antigen bearing cells. And so you have to overcome antigen heterogeneity. Macrophages are well suited to address all three of these challenges. Number one, macrophages are actively recruited into the tumor microenvironment. Tumor cells are producing chemokines, and macrophages have chemokine receptors, and they're drawn in. We see good mobilization of our macrophages into the tumor. Once they arrive, they're unaffected by the immunosuppressive environment. They engage target, and then they start to eat. They start eating through the cancer cell. At the same time, they are producing various cytokines. Our macrophages are polarized to an M1 phenotype and maintain that throughout their life. And they produce cytokines that warm up the tumor. They're able to recruit in T cells, dendritic cells, NK cells, and even convert some TAMs into M1 polarized macrophages. So a net effect of warming up the environment. Lastly, they address antigen heterogeneity through antigen presentation. So as they're chewing through the cancer, they're processing and presenting neoepitopes out to the T cells that we recruited in through MHC, and getting an adaptive immune response for other unique epitopes for that tumor. And so it's a powerful tool, and the biggest challenge, of course, was how to engineer them. And so we are really the leaders in this space. We have identified Ad5F35, as I mentioned, as a vector. We also have exclusive rights to VPX lenti, the only other viral vector that can work with macrophages. We've also spent quite a bit of time chemically modifying mRNA so we can get good antigen or good CAR expression for a period of time through mRNA. So we have a toolbox of gene delivery. We also have the ability to engineer both monocytes and macrophages. Monocytes would then differentiate into macrophages in vivo. And we have the ability to tune the phenotype from an M1 to an M2 and really have that tunability concept well locked in. Lastly, we have the ability to deliver a variety of payloads, CARs, of course, but cytokines and peptides, sometimes transgenes in addition to the CAR. And so anything that you could have a gene for, we can deliver with the macrophage. For the rest of the talk, I'm going to be talking about Ad5F35 transduced macrophages, polarized towards an M1 expressing a CAR. And that's really the setup of our pipeline. The HER2 program, as I said, will be entering the clinic shortly. Meso and PSMA are following on about a year behind. We have another 11 antigens that we're exploring, many in solid tumors, but also some heme malignancies underserved by T-cell approaches. I'm not going to spend a lot of time talking about the data, as this has been previously published. I refer you to the Nature Biotechnology paper in August that talks about our ability to traffic, phagocytosis, polarization, recruitment of immune cells, et cetera. But I will show you some unpublished data. And this is a model that we recently brought up that is able to test epitope spread. And specifically what we did is we took immune competent mice and implanted two different tumor into the flanks of the mice. One is a HER2 positive, one is a HER2 negative. And we know that our macrophages will only eat the HER2 positive. So we treated the HER2 positive tumor with our CAR macrophages targeting HER2. And as expected, and replicating the work that we've seen previously, on the graph in the middle, you see that the HER2 positive tumors untreated, they grow through. Untransduced macrophages, they grow through. But with the CAR macrophages, we see about a 75% CR rate and good tumor control. Now, on the contralateral tumor, we would expect to see similar growth. Untreated will grow through, untransduced will do the same. And where it gets interesting is that the CAR macrophage treated well, the mice that are treated with the CAR macrophages targeting HER2 see a reduction in growth of over 50%. Now, there's no way for the CAR macrophages to actually be eating the tumor mass in the HER2 negative. So we speculated it must be contributed to by other approaches or other cells. And in fact, that's exactly what's happening. We stacked the mice, looked at the tumor, and saw an overall influx in T cells, overall T cell count. CD8 positive T cells go up, CD4 positive T cells go up. And importantly, we also saw CD8 T cells directed at GP70, which is a known epitope in the CT26 cell line, increasing. So we're seeing specific epitope spread against a known antigen by T cells. We also saw, interestingly, increase in both NK cells and dendritic cells. So this is the first evidence that we've shown in our models that we are getting good activation of that second effect, which is the adaptive immune response, and getting specificity for neoepitopes within the tumor. Now, we went further to say, okay, if we're getting this T cell response, what would happen with a combination with PD-1 blockade? And so on the upper graph here, you can see treatment. This is treatment with our untreated, untransduced, or CAR macrophages alone. And then we combined untransduced with PD-1 or CAR macrophages with PD-1. And where we see the great synergy is in that CAR macrophage plus PD-1. And so at this time point, we're out 50 days with good tumor control. And you can see the survival curves at the 80 days here. We're seeing we still haven't reached median survival. So it's suggestive that we are having a synergistic effect with PD-1 blockade. We're continuing to do greater evaluation as the first data that's come out, and we'll present that at upcoming meetings. Now, I do, in the interest of time, want to move over to our clinical program, talk about our lead program, our manufacturing, our phase 1, and what we're going to be looking at. So our lead program, CT-0508, is, as I described, a monocyte-derived macrophage transduced with Ad5F35 to express an anti-HER2 CAR. That CAR is described on the upper right, where we have a SCFB derived from Herceptin, CD8 hinge, CD8 transmembrane, and a signaling domain, CD3 zeta signaling domain, very powerful phagocytic signal. In fact, the most power of all the domains that we've tried. No need for a co-stim at this point. The macrophages are polarized towards M1, and they stay in that M1 phenotype for the life of the macrophage, which we think would be around 3 months or so based on the preclinical work. Now, we've done all of the necessary IND-enabling efficacy and safety work, but I did want to spend just a moment talking about our manufacturing process. What we do is we pre-mobilize donors with GCSF. We get day -4 or so sub-Q GCSF, and then we apheresis the patients and are able to collect around 40 billion PBMCs. We isolate CD14 positive monocytes, and we get around 12 billion monocytes, and then we differentiate into to macrophages using GM-CSF, and then transduce on day 5, yielding around 5 billion CAR macrophages. Those are cryopreserved and then shipped back to the site. Our current vein-to-vein time is approximately 3 weeks, accounting for sterility and mycoplasma release and QA release, but we believe that during the phase 1, we'll get that down to about a 2-week vein-to-vein time. On the phase 1 design, we have planned a multi-center, so it'll be at Penn and UNC phase 1 study to establish the safety and manufacturing feasibility of our program. It's a basket design, so we'll be enrolling HER2 positive 2+ and 3+ overexpressors, so we'll expect to get breast cancer, gastroesophageal, but also HER2 positive colon and lung and ovarian. And so the idea is to get as broad a swath of patients as we can to see what the performance of the CAR macrophages in various tumor histologies. As this is a safety study, for the first 9 patients, we'll be doing intra-patient dose escalation. So we're going to start out and they'll receive the full manufactured dose, but we'll fractionate that dose. So on day 1, they'll receive 10% of their dose. On day 3, 30%, and then the balance on day 5. If it's clear over those first 9 patients, they will then switch to bolus infusion of the full manufactured dose on day 1. Clearly, we're looking for safety and feasibility, but also this is an important study as a first-in-man study to get as much information as we can in other areas. So we want to look at trafficking, which we'll be able to demonstrate through post-treatment biopsies at day 12. But we're also planning to radio-label cells with zirconium-89 and image with PET. And so for the first 30 days, we'll be able to see exactly what's happening with macrophages as they leave circulation and become tissue resident and what proportion are making it into the tumor. We'll be looking at target antigen engagement. We'll be looking at cell killing, tumor microenvironment modulation, adaptive immune response. It's a very robust correlative clinical correlate program that we're working on to get the most we can out of this initial 18-patient study. In terms of our milestones looking forward, as I said, we'll start the phase 1 shortly. We'll start enrolling patients. Hope to get some early safety data by the end of the year. Start getting the biopsy results in Q1, and then start building that data set over 2021. In parallel, we'll be nominating our PSMA development candidate and mesothelin development candidate for moving towards IND. And we're working quite aggressively, I didn't have time to go into it, on various platform enhancements, CAR monocytes that self-differentiate into macrophages, novel constructs, other ways to enhance our overall signaling, combinations with T-cell checkpoints, combinations with CD47, really setting up preclinically what's the best approach for our phase 2 program in 2022. We're also working on a number of CMC enhancements, suspension-based culture to increase our capacity, non-viral gene delivery, et cetera. Again, setting us up for phase 2 development, what makes the most sense. We are currently planning a Series B, and hopefully by the time you're seeing this, we'll close that Series B. And that'll give us fuel to complete all of these milestones we're talking about. So to recap, we're really the leaders in engineered macrophages. We have a proprietary platform, have IP supporting the ability to insert a CAR into monocytes, macrophages, and dendritic cells, both virally and non-virally. We have an emerging pipeline of various agents. We have a GMP supply chain. We can make this consistently. And we have a great team on the ground, great team of advisors, and multiple value catalysts over the next 12 months. We're really looking forward to presenting further information as we go forward. And again, thank you to ARM for letting us present today.